Please use the link below to get the notes, questions and other videos. Don't forget to subscribe, like and share to others for our daily new videos.
So today we're going to look at the importance of meiosis, non-disjunction. How does non-disjunction bring about Down syndrome? And then we wrap up with this topic, and then we can start the topic of reproduction in vertebrates, so that the other time we come back, we can start reproduction in human, or what you call human reproduction. All right. Importance of meiosis. Before we start the importance of meiosis, don't forget that meiosis is the process whereby one cell divides to form 4 daughter cells, and each cell has half the chromosome number to the parent. It means that the cells which are being formed under meiosis are different. So, and then we say that meiosis results in haploid cells, and these cells are specifically for gametes. Based on that information, we can identify some of the importance of meiosis. So, what are some of the importance of meiosis? The first one is the production of haploid gametes. When we talk about the gametes, we say that gametes, these are sperms and ova. So these sperms are haploid. It means that if they are diploid, if the body cell is diploid, it has 46 chromosomes. Now the gametes, they are going to have 23 chromosomes if it is a human cell. So number 2 is halving effect of the meiosis overcomes the doubling effect. How is this halving effect being brought about? When you talk about halving effect, remember that we started with the you start with 46 chromosome. This 46 chromosome, they divide, and when they divide, you form one cell, another cell, and then this one also divide, form another cell, another cell. We want to explain the effect, this issue of halving effect. Now this is 23, this is 23, this is 23, this is 23. So now it means that the sperms or ova which are being formed here, they are going to be 23. Now if you bring about for example, a sperm fuses or fertilizes the ovum, and then these are gametes, remember, then it means that this is 23 and also this is 23. So now when they fuse during the process of fertilization, now you go back to 46. So it means that this is fusion, this is fertilization. So this is the doubling effect. This and this, they double to form 46. But now meiosis is going to divide them now to form 23. That's why here they are saying that the halving effect, this halving effect is being outcomed by the what? The doubling effect of fertilization. So this is fertilization, while this is meiosis. So it means that this helps us to keep the number of chromosomes in one generation to the next generation constant. But if this halving effect was not there, it means that if fertilization occurs now, it would have been now 46, and then you form another one which is 92, then 92 again, 92, again you form a big number. So this halving effect, this doubling effect is being outcome by doubling effect. The halving effect overcomes the doubling effect of fertilization. So meiosis results in reducing the chromosome number. So we are saying that thus maintaining the constant number of chromosomes from one generation to the next generation. So now we are saying that this produces, number 2, another thing is it produces what you call the genetic variation. Remember in prophase 1, remember in prophase 1, you have a process called the crossing over. This crossing over, it helps in exchange of genetic material. So because there is exchange of genetic material, then you will have different sperms. We know that you produce over 300 million sperms per a single ejaculation. So each sperm is different. It's as a result of prophase. During the prophase, in the process called crossing over. So it brings about new recombinants, new combination of genetic material. we are different from our sisters and brothers is because of basically because of the process of crossing over. Why is meiosis important for survival? Do we need this meiosis for survival, or do organisms which don't undergo meiosis survive? We need to this meiosis so that we can create differences among individuals of the same species. If these organisms are known are the same, then automatically it means that there won't be differences. Imagine I asked you a question last time that if everyone is rich, what will happen if there are no differences among us? If everyone is clever, you don't need to read, everyone has the information, it means that there will be no competition. So the reason why meiosis is there to bring about the differences among organisms of the same species. You can survive in hot environment, you can survive in cold environment, you can survive and other ones who survives in hot environment can't survive in cold environment. So it brings about that now all the environment can be occupied by different kinds of people. Yes, if you can't survive, then it means that you have to migrate to the area which you can survive or where you can compete better. If you can't, then you die. You are wiped out. So the importance of meiosis is to bring about variation so that now there is survival of these organisms in the population. So it is a great importance there because it creates genetic diversity in the population, hence difference in the survival of the organisms in the environment. So basically that's what I say, that it brings about what you call survival. From there, let's look at what we call the non-disjunction, the consequences, non-disjunction and the consequences. When you talk about non-disjunction, you need to know where does it occur, which phase. We need to know the phase, Where does non-disjunction occur? So it occurs in anaphase. It can be anaphase 1 or it can be anaphase 2. And then what is the importance of this non-disjunction? Oh, does non-disjunction has any importance, or it has the negative effect on organisms? So that is what you are supposed to cover in there. So we are saying that non-disjunction, each nucleus should contain 23 chromosomes. You have to know that if it is a human cell, this is a human cell, that each nucleus must contain 23 chromosomes after meiosis. But if now non-disjunction occurs, it means that if the chromosomes fail to separate, if chromosomes fail to separate, that's what you call the non-disjunction during anaphase. If the chromosomes fail to separate, then it means that this number of 23 will not be brought about. So one nucleus. So now what happens in non-disjunction? Yes, okay, saying that you have a cell under metaphase, metaphase 1. And then now this cell has chromosomes. Remember, they are supposed to be aligned along the equator. You see, we're explaining the process of non-disjunction. They're supposed to be aligned along the equator. Now, in actual sense, in the normal cell, if it is normal, yes, we expect it to be like in anaphase, I expect it to have these chromosomes separating. You see, these chromosomes must separate. You see, they are separating. And then in telophase, I expect this cell to form two cells, whereby these two cells, they have two chromosomes. You understand? So if it is a normal cell, I expect it to be 46. Now this one is also 46. After at the end of telophase 1, I expect to be 23, and also this one 23. But if non-disjunction occurs, disjunction occurs, what happens? Metaphase, the chromosomes, yes, they will be fine like that. In metaphase, they will do what? They will be fine, yes. And then when they go to anaphase, now because we said it fails to separate during anaphase, yes, these ones are 46. If it's a human cell, then automatically is going to be, now what's going to happen? is going to happen that the whole set, this whole set is going to go to one side. The spindle fiber are going to fail to contract from this other side. You understand? And then it pulls this one, also pulls this one, but this one is going to go on one side. You see, it's going to go onto one side. Now, this is going to result in a cell having, yes, 1, 2, and then 3. 1, 2, and then 3. And then the other one is going to have only one chromosome. You understand? So now, it means that this one is going to have an extra chromosome. This one is going to have an extra chromosome. Yes. And then this one is going to have short, is going to be less of one chromosome. So it's going to have an extra chromosome. This one is going to be less of one chromosome. So now, if this one is still 46, now this one is supposed to be 23, but it has an extra. Therefore, it's going to be 24. And then this one is supposed to be 23, but now it is short of one chromosome. It's supposed to have 2 chromosomes. Now it has 3. It has 1 extra. And then this one is supposed to have 2, but now it is having 1. It means that it's short of 1 chromosome. If we go back to a human cell, it's supposed to be 23, then it means that it's going to have 22. So now what happens? So because now it has three chromosomes, then in science we're going to call it trisome. Tri. We call it zome because it is a cell, it's a body which describes a cell which has 3 chromosomes of the same kind on the 3 chromosomes of the what? Of the same kind. Then here is gonna be, because it is 1, is gonna be mono. Because it's a cell, a body, then you're gonna call it zome. So it's gonna call it monosome. If this non-disjunction occurs on chromosome number 21, then we're going to call it trisome 21. We're going to call it trisome 21. Now, what happens? If this cell, which has 24 chromosomes, the trisome 21, trisome 21, if it fertilizes a normal gamete, for example, this is a sperm, and then fertilizes a normal gamete, which is the ovum. Yes, ovum. Then automatically, what we're going to have is, we're going to have a normal ovum. Remember, it has a normal cell, it's going to have 23. So now it's going to have 24 and 23. Then you're going to have 47. Remember, a normal person is supposed to have 46. Now, instead of 46, it's going to have 47. So it means that now this condition, because of a normal cell fertilizing the cell which has an extra chromosome on chromosome number 21, is going to result in what you call Down syndrome. It's going to result in what you call Down syndrome. That's how Down syndrome is being brought about. However, we can also have a situation, because we say that this cell is having 22 instead of 23. So now if 22 fertilizes the one which is 23, yes, now it means that it's going to have 45 instead of 46. Still, this is also an abnormal individual. So a monosome, if it fertilizes a normal cell, it's going to become 45. And then a trisome, if fertilizes the normal cell, is going to form 47. All this results in abnormality in an individual. So that's what we are trying to explain. So now, when we say that one nucleus of when non-disjunction occurs, as we say that one nucleus will contain 22 chromosomes. We have seen where do we get the 22 chromosomes? It fails to separate, and then one nucleus is going to have 22 chromosomes instead of 23, while the other ones will have 24 instead of 23. It means that this one is a monosome, and then this is a trisome. It is less of one chromosome. It has an extra of chromosome. When either of these resulting gametes fuses with the normal gamete is 23. So now, if this fertilizes this 24 and 23, is going to be 47. 22 and 23 is going to be 45. That's why they're saying that result in 45 or 47 chromosome instead of 46, which is 23 plus 22, you get 45. We have explained that. And 23 and 24, you get 47 instead of what? 46. 23 plus 23. So this will lead to a child having what you call Down syndrome. Down syndrome. And then the child will have 45 autosomes with 3 chromosome number 21. It means that chromosome number 21 is going to be 3 of them. That's why we call it trisome. That cell is called a trisome because they have 3 chromosome number of the same kind instead of a normal pair, and 1 pair of 6 chromosomes. So 6 chromosomes. So if there are 25 plus the 6 chromosome, which is X and Y or X and X, based on which gender is it, then it's going to become 47. And then the women over the age of 40 have a great chance of having children with Down syndrome. We're not saying that children, all the people who are 40 years and above, they're going to produce kids who are having Down syndrome complications. No, we are saying that the people who are 40 years and above, they have a great chance of having this mutation, have a great chance of having a baby with Down syndrome. I think you have ever seen people with the Down syndrome. You have ever seen them. So what are some of the characteristics of people having Down syndrome? Stocky body. The body is stocky. Flattened face with a small and broad nose, small skin. Yeah, you have ever seen these people. Let's look at a picture which has for Down syndrome here. So the folds in the inner corner of the eyes that appear to be slant upwards, and then a large tongue. These kids, they have large tongue. Actually, the tongue is always protruding out. Yes, with the small eyes. Actually, the eyes are tilted. Yes, and then you're saying that the broad hands, the hands are big and short. Actually, the fingers, they are big and short. They are broad, big and short. And then you're saying that wide space between the first and the second toe. If you check you may not see the kid because of the shoes. But if you get a chance, this kid to remove the shoes, you can see exactly what is happening. Yes. So now let's look at some of the differences between meiosis 1 and meiosis 2. Here we say that we explain this, that if you want to know the differences between meiosis 1 and meiosis 2, just look at the phases. Prophase, what happens? In prophase, there is no crossing over. There is crossing over. There is in metaphase, chromosomes align along the equator randomly in homologous pair. Chromosome line along the equator singly. So, and then anaphase, homologous chromosomes separate to opposite poles, while the other one, chromatids separate. Yeah? Telophase, you form 2 chromosomes, sorry, 2 cells. The other ones, you form 4 cells. Crossing over occurs in prophase. There is no crossing over. So you have to look at the phases. Okay, chromosome align along the equator in a homologous pair. You see the word homologous. In the homologous, this is the key point, in the homologous pair. Then the other side, the chromosomes align along the equator individually. This is the key point which differentiate them. Then they are saying that the chromosomes move to opposite poles. They move to opposite poles. They are not saying, they're saying chromosomes. It means that they were chromatids now, sorry, homologous chromosomes. So the chromosomes now, they move to opposite poles. What moves to opposite poles in terms of meiosis 2? Is the chromatids. Chromatids move to opposite poles. Why do we call them daughter chromosomes? They have not yet developed to form real chromosomes. That's why we call them daughter chromosomes, but we prefer saying chromatids. 2 cells form at the end. So we say that here you form 2 cells, while the other side you form 4 cells. You see the key point? Yes. And then our chromosomes number in meiosis 1 is halved. Chromosome, you have the halving. Here, you have the halving effect. You can't see because of the color. And then this side, the chromosome number is maintained. It means that you start with 23 and then you end up with 23. Yes. Then crossing over takes place. We say that there is crossing over, while the other side, there is no crossing over. There is no crossing over. Basically, those are some of the differences between meiosis. What about the similarities? Do we have some similarities between meiosis and mitosis? Yes, obvious. There are some similarities. What are some of the similarities? Both processes involve division. In meiosis 1, you have what called the cytokinesis. Even in mitosis, you have what called cytokinesis, division of the nucleus. So in both cells, you have you have cell division. It means that new cells are formed. However, in meiosis, you form 4 cells, and then in mitosis, you form 2 cells, which are identical. Both process occurs in M phase. M phase is the phase whereby the cell starts to divide. The cycle is divided, into M phase. You have the G phase 1, the S phase, and then the G phase 2. All this is interphase. That's why we call that this is not a part of meiosis. And I can call this growth phase. Growth 1, synthesis, growth 2. And then this is where there is division, the real division which is taking place. Actually, it takes around 80 minutes for it to happen. While this one takes around the remaining from 24 hours. That's what it takes. Yes. All right. In both cell cycle, the stage are common. You have metaphase, prophase, anaphase, and telophase, both in meiosis and in mitosis. There is synthesis of DNA, both. You find the synthesis of DNA in interphase. You see here in the S phase, which you call synthesis. That's where you find the organelles and DNA are being synthesized from. Cytokinesis, division of the cytoplasm. Yes, cytokinesis occurs in the 2 cells. What about the differences between mitosis and meiosis? Difference between mitosis and meiosis. Okay, here we say that it occurs is in the cell body, which you call the somatic cells, while this one occurs in ovaries and testes, resulting in gametes. Production of genetically identical. These ones are identical, while these ones are genetically different. They are genetically different. This one results in 2 daughter cells, while this one results in 4 daughter cells. These cells must undergo maturation, interphase, so that they can mature, so that they form what you call real cells. We call them daughter cells because they are still young. Then you have 1 nuclear division. We don't have mitosis 1 and mitosis 2. No. So it means that the nucleus divides once. That's why we say 1 nuclear. While here we have 2 nuclear division. Here we have crossing over takes place in prophase 1. While here crossing over takes place, there is no crossing over in prophase 1. That's why they are always identical. While in meiosis, there is crossing over which takes place in prophase 1. Then here we say that it results in daughter cells being diploid. You start with the diploid, to N, you end with diploid, to N. While here you start with the diploid, to N, and then you end with haploid, 1N. You see? Yes. Then here results in the somatic cells, where this one results in the gametes. So basically, those are the differences between meiosis and mitosis. So now let's look at, so I think our next class, let it be reproduction. You're gonna look at the reproduction. I know that many people like this topic, and then even in my even in our new YouTube channel for Bright Doctor, we were gonna also discussing too much of reproduction. Yes, check the link below. Don't forget to subscribe and like, and also share to others so that they can also benefit from our classes. Thank you very much. See you again.